Just to shorten the time, I would like Dr. Radiana Dewayani Antarianto, Master of Biomedical Science Ph.D. as the moderator to continue the Biomedical Forum event this afternoon. So Dr. Nana, the time is yours.
Thank you very much, Vika. So I think we will have a very inspiring discussion this afternoon with Dr. Ria Margiana, with Dr. Isabella Kurnia, as they have been very prolific biomedical scientists in Indonesia and also at regional level and international level. But we are looking forward for the um, lecture from Professor Marco Cornelis DeRuiter. Can PMIB share screen for CV of Dr. Ria Margiana as the first speaker of the day? Mbak Adin, can you share screen?
Radiana, do you want to?
No, it's okay. I think, all right. So Dr. Ria Margiana obtained her medical doctor degree from Faculty of Medicine, Brawijaya University, and obtained her master in biomedical science as well as doctoral in biomedical science from Faculty of Medicine Universitas Indonesia. Currently, she is the head of anatomy specialty of biomedical science, master in biomedical science, and research coordinator. She has a research interest of developmental biology, particularly in peripheral nerve regeneration, neurogenesis, fetal development, stem cells, and morphogenesis, as well as anatomy. The next slide, please. She won many awards, okay, including from teaching awards into annual scientific meeting awards. Next slide, and she has published around 48 papers. Okay, she has many research grant as well. Yeah, next slide. With the topic of exercise, physical, peripheral nerve regeneration, and natural substance. So if we look at her articles, okay, she has 48 articles published so far, and the latest article is in injured sciatic nerve treated with curcumin. So this is a very important topic. Dr. Ria, the screen is yours. You have 15 minutes.
Okay, thank you, Dr. Radiana. I think I will start the presentation so we can start right now. Okay. Good morning, Prof. Marco DeRuiter, and good evening for everyone. I think because in Indonesia now it's already at the evening. Okay, first of all, I would like to thank to the committee who has already prepared this meeting, so we can do the meeting right now. And I would like to thank all of the participants who can join us to this meeting. And I would like to thank also to uh, our head of study program, Prof. Septelia Inawati, and Dr. Isabella as our chief of our anatomy department. And thank you also for all of the member or anatomy staff member, and to all our beloved students who can join us to this evening. And also thanks for all the anatomists from all area in Indonesia for coming for this evening. So today I will like to present and also updating the research that have been done in anatomy about the neurogenesis in adult brain induced by peripheral nerve injury. Uh, Although I thought that you are already know me, I will first introduce myself too. I'm Margiana from Department of Anatomy, and as Dr. Radiana said, I am the coordinator of the anatomy concentration in Master Program of Biomedical Science, Faculty of Medicine, Universitas Indonesia. Okay, so this is the aim and the discussion framework my presentation. By the end of this presentation, I would like you have stronger insight of the evidence of the neurogenesis in adult brain that can be injured by the peripheral nerve injury. And I will divide it into 4 topics, subtopics, which are the introduction and the neurogenesis and the peripheral nerve injury, and the last subtopic is the take-home message. So this is the introduction. I think first of all, if we want to talk about topic, we must have a question like, why do we need to learn all of this topic? But I won't answer it right now. But at the end of the my topic, I think you can we together will answer this question. And then I think you have already know what is the neurogenesis, but I would like to make a review. So just to make sure that we are on the same page about the neurogenesis. And as you know, all that peripheral nerve injury can be so many, but I will focus on the sciatic nerve injury because learning this structure, we can learn many things uh, relating with the function, including the sensoric and motoric. And beside that, this nerve actually have a big responsible function in our main reflex, which is the walking function. So here. Neurogenesis. Now, as we know that neurogenesis is actually the process that produce new nerve cells. It can be coming from the undifferentiated neural progenitor or stem cell into mature or already differentiated nerve cell, such as the oligodendrocyte, astrocyte, and others. So a few years ago, many people thought that actually there is no neurogenesis in adult, but this according to the development of the study of the neural system, many researchers already have reported that actually there is a neurogenesis in adult. First area that is reported to have neurogenesis is the subgranular zone of the dentate gyrus in hippocampus, as you can see here macroscopically. And then as you can see here, this is the dentate gyrus, just close to with this structure. And other structures, subventricular zone. And along with the development of the neuroscience, actually, not only in this area, as we will discuss further in the next slide, there is neurogenesis in other area, such as in the spinal cord and in cerebellum that I will give the example of the report. And they are actually reported about the structural changes, including the neuroplasticity. So actually, there are 4 main stages in neurogenesis. First of all, there is maintenance and proliferation of the quiescent neural stem cells, and will be generated into transmitted cells that is going to next process, which is the phase specification. And they will go to be the neuroblasts and going to the next stage, which is they will have differentiation, maturation, and survival of the immature neurons. And then there will integration into existing brain circuit, and then will result as or to be the mature neurons. As I mentioned before, that not all of the peripheral nerve injury will be discussed in this topic. We will focus on the sciatic nerve because it can cause a significant loss either both in sensory and motor function. This is some of study that I have already take a look because it is very important and simple. So they choose sciatic nerve and they make a special method to make a chronic constriction injury of the sciatic nerve, and they use models rodents, and they reported that there is neurogenesis in spinal cord by finding immature spinal cord neurons. And other research reported there is other location which is correlated with other area, which is cerebellum and pons. They reported there is an extensive neurogenesis based on the structural plasticity in adult mammalian, and which may maintain the memory of the basal sensory level, and actually also act as the adaptive mechanism to the changes in sensory input. And then maybe anosmia, I will take a look at this peripheral pathological condition. As we see, this is having so many discussed recently, causing correlated with the this condition of pandemic. What I mean is the COVID-19, and we have already actually we have this discussion in the previous seminar. In this report or research, they use mice, and they reported in the olfactory bulb. They mentioned or they can check there is some migration activity and new neuron findings in olfactory tract. So from this finding, I think we can have stronger insight that neurogenesis probably there is in adulthood. And this is actually our taken from one of our research. We use Sprogdoli rat. We analyzes the motoric function and also the sensoric function. For motoric function, we use spatial measurement, which is we call it as the sciatic function index, and also we analyze the angle that is can be measured in the footprint of the rat. And in this video, we're not only view from the inferior, but also we view it from the right and left side of this model. In here, we will we will check is there any postural changes, because as we see here, there is dynamic change that is probably affecting the ipsilateral side of the extremity. And others reported that there is changes of the knee angle. There is a difference between the left and right side. So we thought that it is there is a postural changes in this model. So CCI induces adult neurogenesis. Actually, we will discuss focus on the spinal cord and cerebellum, but as we can see that for the chronic postural changes, this is posture changes can be a result of the pain avoidance of the model. And then because this postural changes, it will affect the proprioceptive and vestibular sensory system. That if we check further in this area, in this structure that is having connected with this system, probably there is a change there. And in this, as we can see here, there is injured extremity or injured area will send to the primary sensory neurons that, and then going to the spinal second order like this as one of the component in spinothalamic tract. And then it will send, it will cross first to the contralateral side, and then I will send to the higher brain area. As we can see here, why I need, I give an example cerebellum as an easy structure to study, because actually cerebellum, it has
Dr. Ria, excuse me, I think you have 3 minutes left.
Okay, thank you. Okay, so because in this area there is integrator and between descending and ascending, so it will be good as the structure that is to be searched further. So here, from other researcher, they use calretinin and neon. Calretinin actually a marker for immature cell, and then the neon is the mature, and then they unite this picture. So we can see here the immature going from the contralateral to fulfill, or maybe will be differentiated into the mature, which is in here in this area we can see there is a lack of mature neuron, and then we can predict that will be migration and continue with the continue differentiation, and will be at the final stage, which is the mature cells. So from that, after we understand the mechanism, I think we need to know what is factors may regulate the neurogenesis. What is decreased? What is the increase? So this will be very important for us to develop the I think I have actually have a video, but if the time is not very limited, I think we can skip it. So take home message for the neurogenesis in adult can be induced by the peripheral nerve injury, and learning factors that regulate the neurogenesis can be an advantage to develop new therapeutic strategy. That's all of my presentation. Do I have a minute more, Dr. Radiana?
Okay, the final 1 minute, Dr. Ria. Thank you.
Okay, thank you. I will show you the mechanism to have a more clear about the neurogenesis, but it is taken from
the brain is the most complex organ of the human body. It consists of numerous regions that work in concert with each other to provide various functions. There are two regions of the adult brain, the subventricular zone and the hippocampus, where stem cells multiply and create new neurons. This adult neurogenesis has been shown to be important in memory and learning. The brain has a remarkable ability to learn how to discriminate between stimuli. When two sensory stimuli are very similar, an individual is at first unable to discriminate between them. However, with repeated experience, one is able to separate uncommon components from each stimulus. In our lab, we study mice since they have a highly developed sense of smell. Sensory input is received from thousands of receptor neurons in the nasal cavity that send signals to the olfactory bulb. The subventricular zone contains stem cells that produce 30,000 new cells per day that migrate to the olfactory bulb. Once they arrive, the cells first send a process radially outwards, where they branch multiple times to form mature olfactory neurons with a complete tree-like structure. We are able to specifically label this population by introducing DNA into the cells to express a fluorescent protein. In addition, we also express in these cells a special protein that causes them to be electrically active when exposed to blue light. In our experiments, the ability of mice to distinguish between two odors is measured.
Yeah. Okay, I think that's enough just to show the mechanism how is the neurogenesis in adult. Okay, then that's the last of my presentation, and then these are my references. Okay, thank you for the attention. So I will give the time and to Dr. Radiana. Thank you, Dr. Radiana.
Thank you, Dr. Ria. So I guess you are giving us a very broad overview of the science of neuroplasticity in adult brain, as well as the experimental setting to understand or to elaborate those neurogenesis, neuroplasticity further. Is this something your research will move into forward?
No. As long as I'm not focusing on the neuroplasticity, but we will more focus on how is the fate of the cell will be exist after the induce, so the differentiation and I think more focus on the morphology and the function. Not as long as the plasticity correlated. We can study it as the, but we will have a collaboration with other department. I think we will more focus on
Thank
the.
you, Dr. Ria.
Yeah.
Okay, before moving forward to our second very prestigious speaker, I think I would like to invite the participants, all of the students, to actively engage in our discussion. So please, if you have questions, you can raise your hands, you can unmute your mic. You can also write your questions in the chat box if you feel shy to come forward with your questions. Okay, so the next speaker, Mbak Adin, can you share the CV? Actually, I don't think I need to look at the CV, okay? Because Dr. Isabella obtained her medical doctor's degree in 1998 and then pursue her master in biomedical science in Faculty of Medicine, Universitas Indonesia. And in 2009, she obtained her PhD. I think it's from Japan, but from which institution, Dr. Bella?
Hiroshima University.
Hiroshima University. She is currently the head of the Department of Anatomy, Faculty of Medicine, University of Indonesia, and the chairman of E-Museum Cluster. She is also the technical director of Integrated Laboratory, DRC, FMUI, and the head of CGMP Laboratory Integrated Service Unit Stem Cell Medical Technology, Dr. Cipto Mangunkusumo General Hospital, Jakarta. Next slide. Okay, she has won many awards. Okay, particularly she is recently selected innovation in the publication of digital electronic book 111 Indonesian Innovations, as well as in 2018 and in 2017 inaugurated as the best researcher at Universitas Indonesia. Her research interest is developmental biology, stem cell, morphogenesis and regeneration, as well as anatomy. She has published, next slide, 49 articles with the recent publication, which I think she will be presenting today. Comparison of two methods of 2-AAF CCL4 exposure to induce rat animal model chronic liver injury, comparison of two model exposure to induce rat animal model of chronic liver injury, and the dynamic of two protein expression alpha-amine and alpha-fetoprotein during rat liver development. Dr. Isabella, the screen is yours for the next 20 minutes. Thank you.
Thank you very much, Dr. Radiana. Very good morning for all participants, for our distinguished guest, Prof. Marco, our Adjunct professor. Thank you very much for your participation this afternoon. And all my colleagues from Indonesian Association of Anatomists, and also my colleague from Maret University, and also Dean of Faculty of Medicines of University of Jenderal Sudirman Purwokerto, Dr. Fitranto. Please welcome. We are very happy to have you here this afternoon. So actually for this presentation, I'd like to share our ongoing research in our in our department, and we include master degree students in our projects. So for today, I will limit my presentation in the liver progenitor cells biomarker expressions that we also done with the students, the master students. So this expression we checked in during liver development in rats. So this research also in collaboration with Department of Pathological Anatomy, and then Animal Lab facility, and also Diagnostic and Research Center, and the Stem Cell Medical Technology Service Unit, and also the Stem Cell and Tissue Engineering with Dr. Radiana, also the one of member of this unit. Thank you, Dr. Radiana. So actually, maybe people ask why anatomy department choose liver as topic, and also about the regenerations. So actually, after I finished my PhD degree, I just thinking that actually in developmental biology, what we can do in Indonesia. So can I apply my knowledge, for example, in basic biomedical science, in anatomy specialty for our society, for example, for clinical medicines. So I'm thinking that liver injury that progresses into liver fibrosis and ends up being cirrhosis are still become a global health problem, and also in Indonesia. Recent publication in 2020, but it is a survey, epidemiologic survey in the world. The cirrhosis happened in 41.4 million people in the world. And then it can cause death more than 1.32 million per year, means 2.4 percentage globally. And the cirrhosis cannot be cured with conventional treatment. So the last hope is a liver transplant. And but we know that the limitations of the liver transplantations, for example, like limited donors and then immunological rejections like that. So because after I finished my PhD together with Prof. Siana and from Department of Histology and also my team in stem cells unit, we developed mesenchymal stem cell in-house mesenchymal stem cells productions, and I'm thinking how if I use these cells to regenerate liver in chronic liver injury, for example. So now recently the regeneration therapy with stem cell become very interesting because it gives new hope for these kinds of disease. So um, to doing this, I'm thinking that yes, it is good if I try to study this, and then I can not only study about the liver development, but also I can study about the dynamic in the uh, regeneration, for examples, yeah, and also I can give us some uh, um, um, some informations that can apply for clinical medicine, for example. So a liver is a unique organ that has an extraordinary regeneration capacity. So I think it is a good model to study. And then it can induce by the loss of a mass or parenchyma by resection or by injury. And of course, the various regeneration can stimuli as various stimuli, yeah, regeneration stimuli can trigger the different regenerative responses. For example, in this picture, we know that, for example, in a study of partial hepatectomy, yeah, it can give the stimuli of hypertrophy, yeah, if only small amount of the liver is resected. But if a more resection, it can cause the cells of hepatocyte uh, uh, plastic or do a mitosis. And then more resection can cause a dedifferentiation or redifferentiations. So it is also happened in the chronic severe injury that usually become a problem in our society. So in this case, the hepatocyte decrease or lost its proliferation capacity, so it can activate the liver progenitor cells to regenerate the liver. So the question is, does it mimics the process of development in the prenatally? So start from this question, then I continue to study about the regeneration. So does the regeneration process same with the process of development? So as we know that regeneration is a process of how the damaged tissue or organ can recover and get their natural morphology as well as their function. So here, students also have to know that regeneration is not talking about proliferation only or differentiation only or only proliferation and differentiation, but it is also talk about of how the morphology, the natural morphology can uh, back, yeah, regenerate as well as their functions. So I also see this phenomena in the development. So if we talk about the development, it is not only talking about proliferation and differentiation. So let us see about five main developmental processes. As we know that in developmental, during prenatal development, we know that there is a cleavage, that is a cell division. And also there is a growth as the result of the cell division, of course, that is the increase in size. And then there's also a maturations or the cell differentiation, that is the cell become structurally and functionally different. And of course, there is a changes in form that we call as morphogenesis. And the last thing that usually people forget or don't know is that the development also deal with the pattern formation, that is the emergence of patterns. So by knowing this, we now know that development is not only proliferation and differentiation of the cells, and all of this has a deal with the all of this is controlled by the genes. So there is a controlling by the genes. There is a process of development that direct the cell behaviors. So from all this thinking, and then I start to study on the expressions of liver stem cells or progenitor cells during development, and then continue in its regenerations. So I differentiate my, I group my research into three parts. That is, the first is analyze the genes expression patterns during embryonic liver development. And then I will continue after this finish. I will continue to analyze the gene expression pattern during liver injury, and then analyze the gene expression pattern during liver regenerations. So for today sharing, I will share the finished one, that is analysis of the gene expression pattern during embryonic liver development, and the other two, it is now ongoing research. So my research is start by knowing the liver regeneration ability. So anatomically, we know that liver is very complex. That's why it is believed that this very complex anatomy can make the liver have this ability to have a remarkable ability in regenerations. So as we know that liver has a million small lobules like this. This is lobules of the livers. and then has very special architectures. This very special architecture, there is a central vein in the middle of the lobules, and then also the portal triad, yeah, in the peripherals. This to ensure that the blood flows into the optimal contact with the hepatocytes, that make it to make sure that if there is an injury, so it can regenerate easily. So and then I also start to give brief informations about this. So let us back to our understanding in the development of liver. So first, liver is developed in this septum transversum, so around this area. So the liver diverticulum, yeah, actually here, this is the liver diverticulums, originates from the endoderm and develops in the transverse septum, like this. Yeah. So I will show you next of my slides. Yeah. Sorry, I will I will stop this because I want to share this first. Sorry. Yeah. So this is the process of development of initiation, proliferation, and differentiation after the endoderm cells enter the process septum transversums. So this is the process septum transversum. So we check all the histology with SA staging. So it is very simple. Yeah. So we check that start from the embryonic day of rat, start to 12.5 until adult. Here we know that it is slowly, it is the progressions of the development until in the middle, yeah, almost in the middle of the prenatal period, that is embryonic day 18.5, then we can see the lobules of the liver, the radiate morphology, and it so in neonate still not reach all the morphology of adult. So and then we check it more detail. So we know that in embryonic day 18 is maximum in all development, and then it continue in neonates, and it the maturation will still happen in adult until adult. So this is what we study from very simple research. Sorry, I will back. So that is what we can see in very simple observations. And then if we know why this can happen very systematic, yeah, and progressive. And then if we know about the model of regulatory mechanisms of a cell surface marker, yeah, for hepatic epithelial cells during mouse liver development, here in maturations, we can know the maturations of cells. That is, there is a pre-hepatogenesis, there is a specification, there is a proliferation and differentiation and maturation. It can involve so many proteins. So after observe in a very simple way, and then we continue to do a research and try to find a specific marker for these hepatic progenitors. And actually we found so many. So, and then, and also we check the locations, and all the location is, we know this is a central vein and this is the portal triad. So all the the hepatic progenitor cells locate in this area. So around the bile ducts and also the canal of Hearings, yeah. And then, almost all is in the periportal area. That's what we know from our systematic review. And then from here, and then we check which one is the marker for the hepatocyte, for the cholangiocyte, and then for the hepatic progenitor cells. From here, and then we check for the candidate protein that we want to study, so we can know the relation between all these protein with the stages of development. And then we choose these 5 proteins, that is the alpha fetoproteins. I think all of you know about this because it is very famous. And then this is quite new protein, that is delta-like 1 homolog protein. And then this is what we know as adult stem cells biomarker, that is OV6. And then for the CK19 also we know it is famous. And then alpha so albumin or alpha albumin. Why I not choose the albumin? Because it is very early, can expressed by mature hepatocyte. So then I skip this. So after we check with the simple HE staining observation, and then we continue with checking the expression of all these proteins, and we start from the alpha protein, that is a member of the albumin gene family that express in the proliferative cells. So it can express in the progenitor liver or hepatic cells, and also these cells can give rise to both hepatocyte and cholangiocytes. So our research showed that AFP expressions in rat liver tissue begin at the age of 12.5 and reach peak in 18.5. As we know just now, in 18.5 from our observation, we can see there is a peak also in development of the cells. And also it will decrease in adult and postnatally. And we continue to check the expression of the alphamines, and the alphamin as expression in contrast with the AFP. So it is also actually a member of albumin gene family that express in the mature liver cells. So both this expression is like a previous study, other center study. It is we only want to make sure that our system of observation of research is right. So it is start to express in 18.5 embryonic days. And then we continue to check for the DLK1s. Actually, DLK1 is quite new proteins. So this protein is a delta-like 1 homolog protein. It is found during liver development and oncogenesis. So in cancers also it is expressed. So it is a transmembrane protein. It is an inhibitor of notch signaling pathway actually. So this protein expressed in embryonic day 12.5, peak at 16.5, and then 18.5, and then decrease after that. So we see that it is peak earlier and disappear slower than alpha fetoprotein, but they are distributed in the same pattern. So we can see in the pictures the color of brown. It is the positive expressions of delta like 1 homolog. And then we also check that we know as adult stem cell for the liver, that is OV6. So actually it is become our question, my question, whether OV6 also express in during prenatal development, because we know that OV6 only express during adult. That's what we know. But by this research, I know that we know that OV6 also expressed during prenatal development. Yeah, but the different but differ in the locations. So in prenatally, it is expressed in ductal plate cells group, yeah, ductal plate cells, and then but in postnatal liver development, you can see in these pictures, yeah, it is distributed primarily in the biliary duct cells and in the herring canals. So and next we check for the CK19. So CK19 is expression was first seen in 16.5 with a strong intensity in the hepatoblast around the primordium of portal veins. Yeah, here around the portal veins. Yeah, and reach its peak at 18.5, then decrease after that. Yeah. So by observation of normal development, now I try to understand the mechanism of liver cell maturations, yeah, and maintenance of its liver stem cells. So first, the liver stem or progenitor cells, SPC, express IFP like this because as long as we know all the progenitor cells of a liver express the IFP. And then the next, as the time passes, yeah, the stem cells, progenitor cells, undergo maturation and differentiation, and it is characterized by the expression of alpha-amine. And then the maturations can be early seen by differentiations into ductal cells, cholangiocytes that we know by the expressions of CK19s, this yellow one. And then next, how about the DLK1s? Actually, before I think the DLK1 is a marker for fetal stem cell, I think. Fetal liver cell, I think. But here, yes, it is actually expressed in undifferentiated, unmature liver cells. So this DLK, we assume that this protein can maintain cells in immature conditions during prenatal life. And then lastly, for the OV6, actually it is start to express during the prenatal development, and it is maintained in a very minimal expression during adult. So what we can see here, when we see here all the expression reach the peak in 18.5 embryonic development. So it is also in relation with what we observe in the development of the cells, the sinusoid, the hematopoietic cells. How about the development of the central vein? It reached a peak in the 18.5 and then continue to develop until neonates, and it is complete in adult like this. So what we have studied here is that, our candidate protein expression were consistent with the formation and maturations of the liver and the existence of liver progenitor cell populations. So next challenge, actually, some protein that were expressed during development expressed during liver regeneration. This is actually the fact that we can review from other research actually. So for the next, I will continue to analyze the gene expression pattern during liver development with all these candidate protein. And also we will continue to analyze the gene expression during regenerations without the stem cells injection, and with the help or support by the liver cell injection. That is our newest research in um, FMUI, that master degree students can join this project. So this is all my team. So not only from Department of Anatomy, but also include all these persons, departments. Thank you very much.
Thank you very much, Dr. Bella. A bit over time, but I think it's really worth it to see the whole big picture of your liver development research. Okay, it's
Thank
very exciting. Okay, we have several questions at hand. I think before proceeding to Prof. Marco, may I ask a short answer from Dr. Ria Margiana? There are three questions. First from Dr. Yudi Wonowiwoho. Is there any factor that could make faster neuroregenesis process, especially in traumatic brain injury? From Satrio Adi from Gajah Mada University, can adult neurogenesis happen in injured peripheral nerve or enteric nervous system, or only limited to specific area? And from Dr. Agian J. Villano, Pharmaco, do you think the learning capability can improve adult neurogenesis? Thank you. So a short answer to those questions. And also we will invite next session of questions. You can just write it on chat. Dr. Ria,
Okay.
3 minutes, your time. Thank you.
Okay, thank you, Dr. Radiana. Okay, so before, thank you for the questions, Dr. Yudi Yuwono and some from PDIB and from UGM and Dr. Jeffy from Pharmacology Department. First of all, for the first question, let's distinguish between the neuroregeneration and neurogenesis. So in neuroregeneration, there is no new cell produced, even though in one time the neuroregeneration can be simultaneously happen with the neuroregeneration. I will answer for the question, is there any neurogenesis in peripheral nerve? Actually, we have research on neuroregeneration, which is analyze the axonal outgrowth, axonal elongation, but we also analyze the population of the Schwann cell. Even though in here we cannot differentiate whether is it from the new from immature to nature, or already immature or mature, or already mature. But in some research already reported there is immature Schwann cell and there is a population of the mature. So I think there is a neurogenesis in peripheral nerve regeneration. In here, maybe there is the burn of the new cell, specifically for the Schwann cell. And then can neurogenesis can be faster? As I mentioned before, there are so many factors that may affect the neurogenesis. The neurogenesis factor, including age. In younger people, in younger rodents, it will be results in faster. Specifically in duration of achieving better results comparing with the all or all people or all mice. Yeah, and then is there any agent that can make better neuroregeneration? So many research already reported so many additional agent based on growth factor, based on the pro-inflammatory based cell-based therapy, adding to the standard therapy. For example, for standard therapy for the axonomesis is surgery. It can give positive results. Yeah.
Okay, the 3 minutes is up. I think the 3 minutes is up.
Yeah.
You do still have two more questions. I would appreciate if you would attend to those questions via the chat room.
Yeah.
Okay,
Okay.
from UGM and from Dr. Jeffy. Okay, so we have further questions. Prof. Marco, do you want to ask directly to Dr. Isabella your questions? I think this is coming from you, right? Master gene that regulates expression.
Yeah,
Yeah.
thank you very much for the opportunity to ask a question. Isabella, I really enjoyed your presentation. I wondered whether there is a master gene that regulates the expression of your studied proteins in the liver. And I wondered whether it should be the part of your next studies, because is that not the key to study the master genes for regeneration?
Yes, thank you, Prof. So actually, we also thinking about that actually. So first, when I scanning all the progenitor cells like that, we just try to find. First, I only try to find first the cell, the marker, the genes of, for adult, for example, because the objective is actually to know the involvement of the progenitor cells actually. So I choose two candidates in that time. That is the DLK1, that is quite new protein genes, and then the OV6 that we already know. But from here, still I still try to check those expression first. But when I do injury in our animal models, yes, we know that there is an expression of the LK1 increase of the expressions, but not in quite increase significant. For example, very increase because it is not good in regeneration if the DLK1 expression is very high, so it should moderate. But still, I'm trying to find actually what happened in the injury that can induce all these regenerations. So I'm sorry for this moment. I cannot answer your question in well, because I still don't know master of the gene. Maybe there is no one gene only, but there are several conditions that induced by the injury can induce the regeneration, I think.
Okay.
Yeah. Yeah.
Thank you.
Okay. Thank you very much, Dr. Bella. Prof. Marco still have one more questions, but I think due to the overtime of the presentations, can we move forward to your lecture, Professor Marco? Okay, so I would like to introduce Professor Marco Cornelis van DeRuiter. He obtained his PhD with a thesis entitled Development of the Vascular System in the Embryo, a Concept of the Origin Systemic Pulmonary Collateral Arteries at Leiden University Medical Center. And during his postgraduate education and training, I'm looking at like 20s of courses during those years. And he obtained Best Teaching Model winner in Leiden University Medical School in 2018, as well as a certificate of, I think this is basic course for Organist Klinisch Onderzoek. Okay. And then for the academic appointment, from 2019 until now, Adjunct Professor of Anatomy, Developmental Biology, Department of Anatomy, FMUI Jakarta, Indonesia, and full-time professor in clinical and experimental anatomy, Department of Anatomy and Embryology, Leiden University Medical Center. So he is responsible for postgraduate teaching activities of the department, skill laboratory organization, clinical anatomy research, 5 PhD students with external grants and teaching. Professional society membership, membership of the American Association of Clinical Anatomists and Dutch Anatomical Society. Institutional activities and responsibility. Until now, he is a member of Alumni Society of LUMC Committee and member of LUMC Medical Education Research Committee. So he is also member of Review Committee Postdoc of NHS. There is no abbreviation or elongation from the NHS, Professor. Okay, so the total publication is astounding, 517 articles, with the latest publication is published in Disease Model Mechanism in 2020 regarding the disturbed nitric oxide signaling, which give rise to congenital bicuspid aortic valve and arthropathy. Also, human adult cardiac autonomic innervation in autonomic neuroscience 2020, as well as anatomical research to develop surgery in clinical anatomy 2020. So, Professor Marco, the screen is yours for the next hour. Thank you very much.
Okay, thank you very much, Radiana, for the nice introduction. And I also want to thank the faculty for the opportunity to share our data. And I'm really honored to be involved in this master program of your brilliant faculty. First of all, I have to apologize. That is an apology of my wife. She said, may apologize for your hair cutting because in the Netherlands, because of the corona, no longer it is allowed to go to the barber shop. So my hair is a little bit too long. But she said it looks like a disturbed professor, something like that. So
distinguished professor, correct?
Okay, thank you. Yeah,
like a true professor.
Yeah. So I really hope that also you in Jakarta overcome all the corona crisis, and in the Netherlands we are still struggling with it, with still increasing numbers. But saying this, I really like to tell you something about heart embryology, and especially on the outflow tract formation, and because that is really one of the nicest things to do, that is studying the development of the heart. And I hope that I can bring something of my enthusiasm via the Zoom to your rooms where you are, and hope to convince that studying the heart is one of the nicest things to do, and that we are still, and although we are studying it for more than 100 years, yeah, there is still a lot to do to understand normal heart formation. And do you see my pointer? Yeah. Okay. So what is of importance is that when you are studying the heart, most probably you all know that initially you start as an embryo as a two-dimensional plate, and here in pink there is a horseshoe-shaped plate of primordial cells that gives rise to the heart. It is a two-dimensional plate, and then there is a process that occurs, and through which the initial heart is formed. So this is a single heart tube with here the venous pole, and here a single inlet, and here a single outlet. So it is just a straight tube of myocardial cells at the outer layer and in the inner sides endocardial cells. And from this stage onwards, the blood is pumped through the heart. And this is just a single tube, so there is no division in a left and a right side. But later on, you all know that the heart is a complex structure and organ. And as you can see here, we have a right atrium, a left atrium, left ventricle, is here the apex, and here a right ventricle. And in between the venous inflow, you also can see here the outflow tract with a pulmonary trunk and an aorta, etc. So something has happened to become from a straight tube to a four-chambered heart. When we look at the developmental stages of heart development, you can see that, for instance, in the human at day 18 of development, you see that the heart tube arises, and the heart tube arises within 6 days. So within a very, very short time frame. And in the meantime, this heart tube is no longer straight, but it also starts to curve a little bit, to loop, is maybe another better word for it. And it occurs also in that period, and the complete heart tube that is also a little bit looped, that has developed at day 26 in the human. So that means that we have developed this straight heart tube, but then what are the processes, but also the time frame to develop from a straight heart tube into a four-chamber heart with a separated outflow tract. You can see that there is a lot of processes has to occur. For instance, there is a septation at the atrial level, at the ventricular level. There should be a conduction system. Valves has to formate. There should be the development of pulmonary veins, the pulmonary arches and the pulmonary arteries. They should develop with the ductus arteriosus, coronary vasculature. And what you can see here, at day 26, immediately after the looping, all those processes started more or less at the same time. And when you look here, you can see that it is also a very, very short time frame, about at day 43, most of the processes have stopped. That means that at this time here, within 20 days, the complete heart has become from a single heart tube developed into a four-chambered heart. So that means that all those processes, they have to be regulated very tightly in a very, very short time frame. And after this period, if you can see with those arrows, there are indeed still a couple of structures that has to maturate for proper functioning. For instance, in ductus arteriosus, it is present, but it cannot close at that moment as it can after birth. So there must be a maturation. And also the valves, at day 43, they are present, but they have to mature into very tiny leaflets still. And the same is also with the atrial septation. But all the septal components, the valves, and all the vessels, they should be present at this stage. So if after day 43, the structure is not present, or it will not be developed afterwards. And that means that you will end up with a congenital heart malformation. Here I have just a sort of small movie of this straight heart shape, and here you have the venous pole, and what you can see is that during development it is heart, it is bending, looping to the right side, and there are all those venous pole structures as the atria they are developing, but also there is a septation here in the outflow tract. You can see it occurring at this moment. Yeah, and so all those processes are visualized. During this process, with all the formation of the septa, also the heart has to develop from a embryonic peristaltic contraction. As we can see here, it propagates the blood through the heart, it pumps through the heart from the venous pole to the outflow tract, like a peristaltic movement. But later on, first the atria has to contract, and then the ventricles contract from the apex to the atria. Yeah, so it is a complete different situation. Yeah, and for that reason, also there must be a isolation between the atria and the ventricles for the proper propagation of the impulses of the system. This is one of the pictures from I've stolen from one of the embryology books, and it indicates the pre and postnatal circulation. And I really want to demonstrate this picture. That is that the prenatal situation is different from the postnatal situation. And one of the major things is, of course, in mammals who has a placenta, we have, of course, those umbilical arteries and umbilical vein, and they have to close after birth. And all those arteries, you can still recognize them in the adults as ligaments. And but there is a major thing within the heart, that is that before birth, at the atrial level, the foramen ovale is open, and blood can thereby flow from the right side to the left side, and then from the left side through the aorta and to the aorta, descending. But also the blood can flow from the right side to the right atrium to the right ventricle, and then flow into the pulmonary trunk. And there we see that there is a little bit strange situation, that all the blood from the, or almost all the blood from the pulmonary trunk is flowing through the ductus arteriosus to the descending aorta. But why is that? And that is not because there are no pulmonary arteries, because they have developed very early, but the resistance within the lungs of the capillaries it is very high, so blood cannot enter into the lungs. After birth, after the first cry of the child, when he is entering this, the new world, the resistance of the peripheral vascular system is decreased immediately, and thereby then the blood can flow through the arteries and the veins of the lungs. And that means that after birth there is an increase here in the blood pressure within the left atrium, and the oval fossa will close after birth. What does this mean? Before birth, for instance, when the lungs are not developing, or the pulmonary trunk is not developing, or the aorta is not developing, or there is no ductus arteriosus, or there are no pulmonary veins, because of the open foramen ovale, the blood can always flow through the heart in some way, and can enter the rest of the body. So that means that during the embryonic period, if a congenital heart malformation develops, yeah, you can live with it until birth. But after birth, a couple of those congenital heart malformations, like a atresia of the aorta, or an atresia of the pulmonary trunk, or when there are no pulmonary veins developed, yeah, the child will immediately suffer from a problem with the circulation, and maybe he or she can die because of that enormous problem. Yeah. Of course, there are a lot of congenital heart malformations that are not immediately lethal after birth, but hopefully they can be corrected later onwards in life. Yeah. And when we are looking at the heart, and this is for a chamber view, you can see here indicated with those circles where are the mostly found congenital heart malformations. And when you see this, you can see that they are midline, most of them are midline structures. And they have to do with the formation of the atrial septum, of the AV septum, of the ventricular septum, or they also one of the problems are the AV valves, and especially here in the midline, yeah, but also the atrial valves, but also the conduction system, and in some way also the myocardium. Yeah. Moreover, a lot of the connections, yeah, can be also aberrant. For instance, the venous system, yeah, you always have to check a child whether the pulmonary veins are present, and if they are present, whether they are connected to the right atrium, because in a lot of congenital heart malformations, the pulmonary veins can be, for instance, connected to the right atrium. Yeah. And also at the outflow tract, yeah, a pulmonary trunk should be connected to the right ventricle, yeah, and an aorta should be connected to the left ventricle, yeah. But in a lot of cases, it can be in a reversed way. So the aorta is above the right ventricle and the pulmonary trunk above the left ventricle. That is called a transposition of the great arteries. So that means that all the segments should be correctly formed, but also the connections to the next segment of the heart. Yeah? And knowing this, and knowing the time frame and the difficulty of all those structures to form properly, yeah, it is the congenital heart disease. It is about 1 per 100 live births, so 1%. And when we look at the percentages of these congenital heart malformations, you can see that 31% are the ventricular septal defects, the VSDs. Yeah, and the second one is the atrial septal defects. Yeah, there's about 12%. Yeah, and then you also have, for instance, the atrioventricular septal defect. So it is a combination of both processes. So when both processes occurs, you can also enter in a atrioventricular septal defect, and you can see that is also 2%. And most of the other ones are less frequent, but they are also very important because they can be lethal at birth. Yeah. For many years, many researchers have started to study the genes that are involved in congenital heart malformation. And here I have reviewed the genes for instance, the ASD or the VSD, VSD or the AVSD, and you can see that when you produce this kind of lists of genes, you can see there are a lot of regulatory genes in it. So there are a lot of master genes, as for instance, in TBX 1 or 5. Yeah, so that are genes and transcription factors that are inducing cascades of a lot of signaling pathways. Yeah, and what you can see when you look very carefully, that there is not a gene, for instance, especially for the VSD. For instance, GATA4, yeah, is also of importance for the formation of a proper atrial septum. Yeah, and what we can also see that NKX2.5 is a gene that is described in the formation of myocardial differentiation. Yeah, apparently it is also involved when this gene the expression is disturbed. You can end up with an ESD, but you can also end up with an AVSD or with a VSD. So that means that a lot of those genes are not cannot be specifically related to one heart malformation. And why is that? And thereby we have to study the embryology. And one of the first things I want to emphasize is that the heart consists of an enormous amount of various cells that contribute to proper heart malformation. So, for instance, we have, of course, a myocardium and an endocardium. So the outer layer of the heart here and an inner layer, yeah, endocardium. Yeah, but there are also, for instance, an epicardial layer. Yeah, the epicardial layer is the outer layer outside the myocardium, but at first stage it is not present, but later on it will grow over the heart and gives the coronaries, but also it is of importance for proper myocardium development. Yeah. Moreover, there is, for instance, cushion tissue. Cushion tissue that is an area in between the endocardium and the myocardium. And here in this electron microscopy picture, I hope you can see it. It is almost empty, but it is empty because there is a lot of cardiac jelly. So it is an enormous amount of jelly-like extracellular matrix. So, and you can see it here. It looks like real pillows and cushions, yeah, and those cushions can deform a little bit. So they are of great importance for the formation of the valves. Yeah. So these endocardial cushions, very specialized processes is occurring. For instance, the endocardium, yeah, has to transform into mesenchymal cells, yeah, to fill the ultimate cells within those cushions and for proper valve formation. So there is an epithelial mesenchymal transformation is occurring in these areas, very specialized processes. Yeah. Moreover, there are also, for instance, neural crest cells. That is an extra cardiac contributions to the heart, and it is essential for proper septation of the aorta and the pulmonary trunk. And of course, you also have endothelial cells and smooth muscle cells, pulmonary trunk. Yeah. So, and what we can see is those master genes, yeah, they are regular. They are not only involved in one structure, so not only in the atrial in the atrial septum or in the ventricular septum. No, they are expressed within, for instance, endothelial cells or in the myocardium, in the entire myocardium. So that means that a lot of those genes, they are involved in many of those processes, from a single heart tube to a four-chamber heart. And thereby, what you can see is that a lot of deviations can occur. So most of the time, it is not only one malformation, but it is always, in most of the time, it is a combination of, for instance, a VSD and in combination with a transposition of the great arteries. So, with other words, it is a spectrum most of the times of the congenital heart malformations within one patient. So, what are the developmental background for it? because that is the major point of my presentation. So we first have to think about the heart from as a single heart tube, and this is a frontal view, and this is a lateral view of the heart, and this heart here is also longitudinal section. So what we can see is here the myocardium, the brownish color, and here at the inner side, the light blue is the cushion tissue. So the future valves. Yeah, so this is the heart, and here we have the head of the embryo. Here the otic placode for the ear, and here in green is the foregut. Yeah, and what we can see is that this initial heart tube, yeah, that is formed from the so-called first heart field. So that is the two-dimensional plexus is remodeling into this. This is the first heart tube. So the first myocardium. After the formation of this tube, when all the other processes of atrial septation and the outflow tract septation, yeah, during that period, there is new myocardium added to the heart. That new myocardium is not inside the pericardial cavity, as indicated here. No, it is outside the pericardial cavity. So that means that this yellow part, this is mesenchymal structures outside the heart region, they can add to the heart itself. And that is called the so-called second heart field. So the second phase of heart malformations. And here, anteriorly in the embryo, this part will contribute to the outflow tract region. But also at the posterior part, yeah, we also have a lot of second heart field that is adding to the inflow tract of the heart. Yeah, and next to those yellow second heart fields, we also have the neural crest cells from the ectoderm here at the back of the embryo, and they are really migrating towards the heart. Of course, the neural crest cells, they are of great importance for the development of the autonomic nervous system of the heart, yeah, but also they contribute to the outflow tract formation, and they form mesenchymal cells, and they are of importance of septation of the aorta and the pulmonary trunk. Yeah. Okay. For many years in the previous century, yeah, it was not possible to study the heart on basis of expression of genes and proteins. And for that reason, all those old studies, yeah, they were looking for specialized areas which we could study with general histology stainings. For instance, it was possible to recognize a specialized myocardium in the area of the AV canal. And this is this purple, or sorry, this blue ring, that myocardium is a little bit different. There is also a smaller area than the atria and the ventricles. So this part of the myocardium was called the AV ring. So it indicates the AV canal. And also in the outflow tract, specialized areas could be determined, and also in between, yeah, the two ventricles. And so here we have the lobed heart with an atrium with a single AV canal. So this is the future tricuspid orifice and the future mitral valve orifice, and all the blood flows immediately here into the future left ventricle, and then the blood flows through this narrow ring, and this is also called the primary ring. It flows to the right ventricle, and then it flows through the single outflow tract. Yeah, and by studying all those specialized areas, researchers were found that those areas, you can trace through development, and you can then see that this area here, for instance, this blue one, is still present here in the adult situation at the level of the mitral valve, the tricuspid valve. Yeah. Also at the outflow tract, you can still see some of those areas, specialized tissue, which could be the lineage of this ring, which was situated over there. Yeah. And what you can see, and I will not discuss it later on, but here this yellow ring, yeah, is a simple ring, yeah, and then later on it curves in a really strong way, and it divides, and it gives formation to the septum and to a part of the right ventricle. So that is a very strange tissue, and you can imagine that if this ring is not curved in a proper way, you can end up with a lot of problems at the inflow tract of the right ventricle. For instance, the Epstein abnormalities, with a problem with the tricuspid valve has an origin in this yellow ring structure. Yeah. And of course, there is all those formations of those rings, yeah. But in the end, also the atrial septum, ventricular septum, and the outflow tract septums has to become of great importance for the proper septation of the heart. So I want to demonstrate you two things, and one is the atrial septation. That is the part of, and which is quite easy to understand how the septation of that area occurs through the second heart fields, and then I want to focus more on the outflow tract. So we start here with the inflow tract, and what you can see here in this movie, we have here the systemic veins of the embryo entering here the future right ventricle. Here we have the left atrium, yeah, and here is the AV canal, and here this is cushion tissue, the future part of the tricuspid valve and the mitral valve. And with the formation, here we have first the atrial septum, the primary septum, and here in green we have the secondary septum, and then we also have some gaps here through which ultimately the blood flows. And here in a couple of stills, and what you can see is that there are two septums, a septum primum and a septum secundum, and those septa, they are formed, and through these gaps, through these foramen, the blood can flow from the right to the left side. Yeah. What we now know is that a lot of those structures will not develop properly, so you can end up in the end with an open foramen ovale, or you can have here, and when the leaflets are not well developed, you can end up with an ASD, for instance a type 2, and when those areas are not overlapping. So then, for instance, when this foramen is much larger, you can end up here with an open foramen ovale. Yeah, and of course you can also see that the disturbed atrial septum defects, and you can have a primary defect or a secundum defect, and here are the genes that are involved in that. And what is of great importance, that all those structures here also here indicated in yellow, yeah, these structures are from the second heart field. So when the second heart field is not developed properly, you will end up with all kinds of atrial septal defects, primary defects or also secondary defects. Yeah, and when we look, for instance, here in the embryo, then we see here that this is a mouse embryo of 2.5 days. Here we have the atrial septum, ventricular septum, and a right atrium, left atrium, mitral valve, tricuspid valve, and here the left and the right ventricle. Yeah. When we compare it with this situation over here, yeah, you can see that here there is a inflow tract of the left ventricle. So all the blood from the right and the left atrium is flowing into the left ventricle. But in the end, the right atrium should be above the right ventricle, and only the left atrium should be above the left ventricle. So, with other words, this blue ring should be moved above the right ventricle. Yeah. If not, you will end up with a congenital heart malformation. Yeah? And moreover, here, the central part should be here also septated properly. And here you can see it here in the embryology. You have not to understand it in detail, but here is the AV ring. So it is just a ring-like structure. But here in the midline, it is first a one circle, and then it is by condensed tissue. It is closer in the midline. So then you have a kind of this is a zero-like structure, and this is an H-like structure, because it is glued here in the midline, and everything is because of the second heart field here, just in the midline. And if this is not occurring properly, you will end up with a common AV valve, because these structures are not gluing here in the midline to the AV cushions. Yeah, and thereby you will end up with a large common AV valve. Yeah, and that can be complete or incomplete, so there is an enormous variation. And for instance, here this is a child which was suffering from a complete AVSD. This is the ventricular septum of the left ventricle. This is the free wall of the left ventricle. This is the apex. So it is we have opened the heart from the apex to the basis of the heart. Yeah, and here you can see there is it is called a bridging leaflet. This is a leaflet that is extending from the left ventricle towards the right ventricle, which is situated here behind the ventricular septum. And here you also have another bridging leaflet. So two leaflets that are completely running from the left ventricle into the right ventricle. Yeah. And what you also can see is that by the second heart field, there is also, and that is for cardiologists, echocardiographist of great importance, is that the mitral valve is always a little bit higher than the tricuspid valve here in the midline, and that is caused here because here is there is no myocardium, but this is the center part of the heart. This is where all the structures, all the fibrous tissues of the valvular leaflets is formed over here, just in the midline. And this is also called the membranous septum. So from the right atrium, you can, without touching myocardium, when you put the needle here and through, here you can, through the fibrous septum, you can enter here the left ventricle. I will skip these pictures of ventricular septation, because they are, although they are beautiful, but what we can see is that there are a lot of muscular or perimembranous defects here within the ventricular septum. And I only want to demonstrate this because the ventricular part of the heart is a very, very complex situation, as there is not one ventricular septum. No, there are four. There is one inlet septum, there is a primary septum, a membranous septum, but also the outlet septum. All those parts, they are essential in a proper ventricular septation. And knowing this, that when there is some one of these components is not developing properly, you will end up with one of those types of ventricular septal defects. Yeah. So. And now finishing my presentation on the outflow tract. And what we can see here is the outflow tract is a single outflow tract. And here you will end up with a pulmonary trunk and an ascending aorta. And how is that occurring? We know, and I already introduced it, that for many years, we believed that the neurocrest cells, that were the only cells that can septate the outflow tract. And they are of great importance. And they are studied very well in the previous century, from the 90s year onwards until now. And what we can see here, when you have a single outflow tract, so this is a transfer section through this area, yeah, it will enlarge. And here, this is the myocardium, this is the endocardium, this is the cushion tissue in between. And what you can see is that cushion tissue is enlarging, it is bulging into this area. And then here in the midline, neurocrest cells, they are entering. And then after entering, they will invade, yeah, they give signals towards the myocardium, through which this myocardium is entering here this area, and then you will end up with a septated pulmonary trunk and an ascending aorta, with also here the three semilunar valves in the, yeah. So that is the single explanation. Yeah. Just to give you another example of the neurocrest. Yeah, the neurocrest are from ectodermal origin. They are start here with their migration, and then here indicated in blue, they are around here the dorsal aorta, and they are also moving here towards the heart. And here they have to septate in some way here the outflow tract of the heart because of their invasion. Yeah. How do we study that? In the previous century, we did it with, for instance, with retrovirus tracing. So you use a retrovirus with its replication incompetent, but it integrates in the DNA of all the ectodermal cells. And because there is a LAC-Z gene also introduced, all these cells, all those ectodermal cells, but also the descent, so also their daughter cells and their daughter cells and their daughter cells, they have recombined, and then there is a beta-galactosidase expression, and you can in a section you can stain it for beta-galactosidase, and you will end up with a blue precipitate. And you can see it here. We have here the pulmonary trunk with here the three leaflets. Here we have the orifice of the aorta with the three future leaflets. And in between here, we see those blue cells, and this indicates all those cells, they are of neurocrest origin. And what you can see here, you see a kind of invasion over there. And here, this is in a later phase, and you see here enormous amount of myocardium, and you can still here see those neurocrest cells. And there is a lot of evidence that this invasion, yeah, is caused by the cell death of those neurocrest cells. Because these neurocrest cells are dying, then growth factors are gives the signals from the cytoplasm, and through which those myocardium is migrating into the cushions and are involved in proper septation. Yeah. Nowadays, we are now using the so-called Krelog's recombination system. I will not go into the details of it and how to produce these mice, but what we are doing is by using this system, you also can trace cells of a specific origin, and you can trace their lineage from embryo to in the adult phase. And so we have a mouse in which we can trace the neurocrest cells, and what we can see here, this is the aorta, this is the right ventricle, and that here we have the three leaflets of the aorta, and what we can see here is those blue cells are from the neurocrest cells, and they are here at the attachments of the leaflets here in the arterial wall. you also can see that within the leaflet there are also some neuron crest cells. And what is of great importance, that not all those leaflets have the same contribution from the neural crest cells. Exactly. So this leaflet has more neural crest cells than those leaflets. You can count and you can prove it. And also here you can see here the remnants of the aorta pulmonary septum, this part here. And later on, for those ones that are thinking, okay, what will be the future structure of it will be the tendon of the Darrow for the specialized morphologists of the heart. So when there is a deficient septation of the outflow tract, you can end up with a common arterial trunk, or also called a truncus arteriosus persistens. And it is a defect at least at 3 levels. And first, it is a defect at the level of the aorta and the pulmonary trunk. So at this level. But it is also at the level of the semilunar valves. So there is not a proper aorta and a pulmonary valve. Yeah? And there is also a communication between the left and the right ventricle. So there is also ventricular septal defects. And how can you organize no, that's the wrong word. So how can it that one structure, a outflow tract structure, yeah, end up with a septation at 3 levels? And I will enlarge this picture over here. This is a child with the so-called 20q11 deletion syndrome. And here we have the ascending aorta, but here we have the common atrial trunk. So this is aorta, but this is pulmonary trunk, but it is not septated. And thereby, we are, so this is a common arterial trunk. And from this here, we see that there are 2 pulmonary arteries are arising from this area. And a little bit lower to that, we can see that here is the common valve of the common arterial trunk. So you cannot see a differentiation between a pulmonary valve and an aortic valve. So because the septum should arise from here, towards here, through the level of the valves, and should meet here the myocardium in between the right and the left ventricle. So that means when the AP septum, yeah, which has to septate this complete area. Here, imagine that, for instance, in a normal situation, it should enter, grow until here. But if it only grows from here to this level, you will end up here with a ventricular septal defect. Yeah. But you can also think about, okay, the AP septum is growing from here towards here. Yeah. So there is an ascending aorta and a pulmonary trunk. Yeah. But there is no septation at the level of the valvular leaflets. Yeah. And that is another. And then you have the possibility that it is not present, and you will end up in this situation. So you have just by the extension of the outgrowth, and the way it is disturbed, you can end up with 3 variations of common arterial trunk. With the really minor thing is the ventricular septal defect. Yeah. So a lot of studies we and others have performed because we all believe that the deficient migration of the neural crest was sufficient for the outflow tract septation. And from human studies with children with the so-called DeGeorge syndrome, or the 20q11 syndrome, as it is now called, it was found that the TBX1 gene, yeah, was mutated. So there was no expression of TBX1, and that is an important transcription factor. So when you want to study the expression of the TBX1, you would expect, because of the outflow tract septation problems, yeah, you would expect that the TBX1 will be expressed here just in the midline, in between the aorta and the pulmonary trunk. But it is present here around the pulmonary trunk and around the aorta. And it is not the place where the neocrest is situated. And in the knockout situation, yeah, in a mouse, you can see that there is completely around and there is some building over there. And we found that the TBX1 is not expressed in the neural crest. No, it is expressed in the second heart field. And when in the knockout, it gives you a complete spectrum, yeah, of outflow tract abnormalities, tetralogy of flow, pulmonary adhesion, common arterial trunk. Yeah. So if it is in the second heart field, there must be signaling pathways between the second heart field and the neural crest cells. And in the end, the neural crest cells will migrate, their migration will be disturbed in that. Yeah, and what we and also a lot of others have studied here that TBX1, for instance, regulates the development of the neural crest cells by retinoic acid signaling. So there is a lot of crosstalk. Yeah, and I like to present one of our models of also the common arterial trunk, and that was in a mutant with the LRP2 knockout mouse. And what is the LRP2? It is the lipoprotein related protein 2. And that is a receptor, yeah, and it is always on the apical surface of cells. It is of great importance for cholesterol transport, vitamin B2. There is a co-transporter with cubuline, and it is also crucial in the cellular uptake of retinol binding protein. So thereby it is also indicated that it is essential for the signaling of retinoic acid, as also in the previous article is indicated. And it also regulates sonic hedgehog signaling, which is of great importance of pattern formation within the embryo. Yeah, and what we found is in these LRP2 knockout mice, also called the megalin protein, is that 8 out of 9 knockout mice, they end up with a common arterial trunk. So this is a reconstruction of a wild type embryo. This is a right view on the heart, and what we can see here is the ascending aorta, and here we have the pulmonary trunk. Yeah, here in the embryo, and this is the ascending aorta, aortic arch, descending aorta, and from the pulmonary trunk there are two small, tiny pulmonary arteries. They are rising from it. Yeah, but all the knockouts look like this, or almost like this. That there was no septation. There is one common arterial trunk and two tiny pulmonary arteries arising from this common arterial trunk. And you can see it over here in the sections, and these are sections. They are stained for myocardium. This is the left atrium, right atrium, and here we have the ascending aorta, pulmonary trunk, right ventricle. You can see here in a wild type there is a septation, but here you can see this is the right ventricle, one common leaflet and one common arterial trunk. Yeah, but what you also can see when you look at, for instance, here at the myocardium, yeah, you can see there is a thick ventricular septum, there is a thick right ventricle and a left ventricle myocardium of the free wall. But you also end up with a lot of problems here within those myocardium. And you can see it over here. You see that it is very tiny trabeculated myocardium. So there's also something wrong with the myocardial formation. Of course, you can look at all the cardiac malformations, and you can describe that very nicely. But what we found was that also in this case was that there was a problem with the second heart field. And these are, I will show you a couple of reconstructions of the here at the left side is the wild type, here is the knockout, and in the red is the lumen of the ventricle of an embryo of 10.5 days mouse development, and here in blue are the cushions, and here is the pharyngeal arch system, with in green here the neural crest, and here in yellow are the second heart field cells adding towards the heart. And in the wild type, we see that there is a nice line of neural crest cells here adding to this part of the heart. But in the knockout, you see that is a little bit at this side disturbed. So you see it is present, but it is not in one beautiful line. The neural crest cells, moreover, they are just entering the heart. They are just nicely entering. When we look at the same embryos, but then from a different view, you can see here a neural second heart field, and here also second heart field, and as a long line. But in a knockout situation, you can see that it is completely disturbed. It is situated in another way, and also at a later stage, you can see that there is something wrong between the knockout and the wild type. Yeah, and when you study these all those stages, you can conclude over there that when we have a outflow tract here with this line in a wild type, yeah, you can see that this here it is gray line, that is the myocardium of the primitive heart tube, and here in yellow we have the second heart field. Yeah, and this second heart field is adding to the heart, and you can see it over here. Yeah, and thereby there is a rotation of the outflow tract, and it is contributing here to this area of the heart. Here, so you see here this becomes all myocardium, but also part of it, because of the rotation, it gives a division of the pulmonary trunk over here. There's in between the two pulmonary arteries. Yeah? Because of this process here is occurring, the neural crest cells are entering at the right place, here in the midline. And thereby there is a septation of the aorta and the pulmonary trunk. But in a knockout mouse, yeah, we can see here, we start with the same situation. The contribution to the right ventricle is not occurring. So this part is not occurring. And thereby there is no rotation. And because there is no rotation, the neural crest cells do not end up here in the midline. No, they enter at the wrong side, at the pulmonary side, at least. Yeah, and thereby they miss their definite position, and there is no longer a septation. Yeah, and I want to demonstrate you all the effects of outflow tract septation with 2 movies, and I hope it will become clear how the outflow tract is formed. This is a young situation with here in red is the ascending aorta, and here is the pulmonary trunk in blue. Yeah, it is a view from above and behind. Here's the AV canal, and this is the in yellow the right ventricle. All the vessels are above the right ventricle. And now look what is happening. It grows, it grows, it grows, and then here, look at what is happening here with the pulmonary trunk. In the at first instance, the pulmonary trunk is situated dorsally or posteriorly to the ascending aorta. But then, by the addition of second heart fields, it moves around the ascending aorta via the left side and migrates anteriorly to its ultimate anterior position. Because you all know from the adult situation, that the pulmonary trunk is in the end situated in front of the ascending aorta. So that means that by this process, by adding of the second heart field, we also call that the pulmonary push. That when this is not occurring properly, you can end up with a pulmonary trunk that is situated here, for instance, next to the aorta, or it remains situated behind the aorta. And that means that we can end up with a lot of abnormalities. And what you can see here is just a picture to impress you. This is a mouse embryo, a four-chambered heart. This is the right atrium. So black is the lumen of the atria and the ventricles, and here in blue is the myocardium. This is the right atrium. This is the left atrium, and here is the left ventricle, and this part here in red is the right ventricle and the ventricular septum. And we have developed a mouse line, and this mouse line is specific for the anterior second heart field. And thereby we were able to determine that the outflow tract of the entire right ventricle and the ventricular septum is of anterior second heart field. And also the outflow tract around the aorta or ostium, so of the area of the aortic valves. And that means that the left ventricle is of the primary heart tube, and this is from the second heart field. So everything in blue. And that means that there is something strange happening, because here, at first instance, above the right ventricle, both the aorta and the pulmonary trunk are above the right ventricle. But in the end, only the pulmonary trunk should be connected to the right ventricle. And with those pictures I demonstrated with that short movie, when there is not a proper rotation, you can end up with a so-called double outlet right ventricle. So the aorta is still above the right ventricle. So a kind of primitive position. It is the initial position that the aorta is above the right ventricle. But you can also have this kind of position. This is a view of the right ventricle, and you can have here in an anterior position the aorta ostium, and the pulmonary ostium is behind. And this is a so-called position. It is also a double outlet right ventricle. So two vessels are connected to the right ventricle, but they are in an initial position. So the aorta is still anterior, and this is in the posterior situation. So there is almost no contribution of the second heart field to the right ventricle. And you can also have another kind of position as discordant. I will end up my story, and I already indicated that there are many genes involved in information of various processes, and thereby you can end up, for instance, with a GATA4 with a ventricular septal defect, but you can also end up with a double outlet right ventricle, and that is caused that it is not only one structure, but as the second heart field is involved, it gives rise to many structures, and many abnormalities will be developed from that. And I really want to end up with the epicardium, And what we can see here in red is the myocardium, yeah? And all the epicardial cells, they are yellow. And we all know that the epicardium is at the outer layer of the myocardium, but through development, you can also see that the lineage of those epicardial cells, they are migrating into the myocardium. And because they are migrating here into the myocardium, the tiny trabeculated myocardium over here, they are invaded, and thereby we will develop a proper myocardium. Yeah? And a lot of studies have been performed how that processes can occur, but they are of essential. Those epicardial cells, when they are migrating into the myocardium, they are forming interstitial fibroblasts, yeah, and they are essential for the proper myocardial maturation and over there. And what we also saw in the in the Magellan knockout mouse, that the knot was only the outflow tract from malformation, but it also is involved in the epicardium migration. So thereby you can also see that a common arterial trunk can coincide with a cardiomyopathy of the ventricle. So my last slide here. So here, what we have seen here, that in the heart, it are mostly the midline structures, and when you have a abnormal looping, so at the first heart field, you will end up with very, very primitive hearts. Most of those hearts will die within the embryo itself, and then you end up with a double inlet, double outlet right ventricle. But when there is something wrong with the anterior second heart field, you will can have a transposition of the great arteries, double outlet, truncus arteriosus, all that kind. And I indicated, but also the posterior second heart field, and that are different from the anterior, but it is can give rise to the atrial septal defects, abnormal venous connected even sinus node diseases, etc. And the NeuroQuest, they are specifically involved in the maturation of the ductus arteriosus, the hyperplasia of the aorta common. It is involved in the truncus arteriosus, although also the anterior heart field is also involved in the truncus arteriosus persistence, etc. And the epicardial cells, they are of great importance for proper compaction of the heart, but they are also essential for the development of the valves. So, deficient looping and malalignment of septal components, yeah, they can explain the defects, abnormal, absent fusion of endocardial cushions, as we have seen, for instance, in the AVSD, and deficiency of the second heart field and NeuroQuest contribution. They are both of essential for proper outflow and inflow tract abnormalities. Yeah. And the hyperplasia of myocardial septum components, and then we are dealing with problems within the myocardium itself or in the epicardium. I thank you for your attention, and I think I have overwhelmed you with a lot of information, but I hope that you have seen and that to study the heart development, and you will study a lot of various processes, and you have to focus on one of those lineages, and then you will find a clue for a couple of congenital heart malformations. Thank you for your attention.
Thank you very much, Prof. Marco. So I hope we still have some time because we are also filled with questions here, Prof. Marco.
Yep.
Okay, from anatomy master student, Dr. Raditya Alfarabi. He wrote, "Hello, Prof. DeRuiter. Prof. DeRuiter, may I ask you, as I know stem cell transplantation for congenital heart disease may possible as an adjunct to surgical therapy and may provide benefits than surgery alone. in terms of somatic growth, especially pluripotent stem cell. Could you please share to us about the roles of stem cell in congenital heart disease that you may have conduct on those projects? Thank you. So that is the first
Yeah.
question. Shall
Okay.
I read some more or one by one?
Just first this question. Um, um,
Regarding stem cell application.
Yeah, one of the major problems is that that the heart after in the adult is cannot divide anymore. So there is no cell division anymore, and the in the adult heart are almost no myocardial progenitor cells. So that means we are very, very unlucky within the heart. So a myocardial infarction will end up with less myocardial cells. So there is no potential over there. And so we have to deal with all kinds of other techniques as induced pluripotent stem cells to add to the heart, and to replace those cells. So it is, for instance, we know from the zebrafish that until day 4 after day 4, the heart can no longer regenerate. Before it can regenerate. So it can regenerate without any scar. In the mouse, it is until day 12. So until day 12 development, all the cardiomyocytes, they are plastic and they can rebuild the heart without any scar formation. But then after that, they lose their possibilities, and that is a real problem. But it is great stuff to study.
Okay, the next question is from Dr. Ria Margiana. So Dr. Ria Margiana, she asked about difference in ASD manifestation if transcription factors like NKX 2.5 and GATA4 are mutated. I think she means using zinc finger factors or the experimentation mutation using zinc finger factors, I think.
I do not know whether I understand the question correctly, but there are a lot of zinc finger problems, proteins problems, all ending up with congenital heart malformations, but I do not know what the question really is. Can I see it also in the chat or?
Yes, Prof, it's in the chat. Or Dr. Ria, would you comment a bit on your question? So are you referring to mutation of the NKX 2.5 and GATA4, or are you referring to the experimentation of the mutation using zinc finger? Which is the direction of your question?
Yeah, okay. Thank you, Dr. Adiana. What I mean is there any difference in ASD manifestation between, for example, the mutation on NKX 2.5 and GTA4?
Oh, okay. Yeah. Now I understand. Not in the morphological way, but it is more phenotypically also in the myocardium. So what you can see is that you will end up with an ESD, and it is always it can be an ASD type 1 or ASD type 2. It does not matter whether you have ASD NKX 2.5 or the GATA4, but I think what we have to think about, the NKX 2.5 is of great importance for proper maturation of the myocardial cells itself. So if there is something wrong in the NKX 2.5, you will end up with other associated side effects than with the GATA4. So I think you always have to know what is the genetic background of an ASD. In former times, we were just only surgeons focused on the ASD, and so we will repair. But nowadays, what we know from those transcription factors, can also influence later on the maturation of the cells, and there it can be a difference. Yeah. So it is of great importance to know it for the long time difficulties in congenital heart malformations.
Thank you, Prof.
Yeah.
Okay, there is this, I think this question is also correlated to it. Questions from Dr. Ria. Well, this is about pharmacological intervention, Prof. Since all of those
Yeah.
cardiac malformation, there are several established genes for this. Dr. Jeffy, he's from the pharmacology department. He was wondering if there is intervention strategy, a pharmacological intervention that could affect those genes and improve malformation.
Yeah, again, a great question. Because you want to interfere. We have 1% of the children is born with a heart defect, and can we interfere with it? I think at this moment not possible. What we know is that a lot of medicines, they are really affecting proper development of the embryo. So you have to take you should be very anxious, I think, not to interfere with it unless you really know whether it is safe or it is only affecting one cell group or something like that. But I think it is much too complicated. That is one thing. And the other thing is that most of those heart defects, you can diagnose them after you can visualize them. So you visualize them at, for instance, week 12 or 15 of the pregnancy, and then you already have the abnormalities because they have developed before 12 the 12th week. So it is difficult to interfere because you will not know it at that moment when they are developing. Yeah.
Okay, Dr. Jeffy, do you still have any further questions? I can allow you to question directly, maybe for 1 minute. Dr. Jeffy? Okay, then we move forward to the next question from Dr. Isabella. Do you think the position of the pulmonary trunk to the aorta is mostly because of the influence of the development of the second heart field, or is it also because the process of septation? So this is very morphogenesis question, Professor.
Yeah. I think that the position of the pulmonary trunk is mainly determined by the second heart field. If the second heart field is not present, you will see that the pulmonary trunk will not rotate properly around the aorta and becoming into its anterior position. And neurocrestals are only necessary to septate. The septation is really because of the neurocrestals, because when you have no neurocrestals, you will end up with a abnormal septation or no septation. But if the second heart field is not developing properly, you can have a disturbed migration of the second heart of the neural crest, and thereby they come to a wrong position and also hampers a septation. Yeah, so to answer it very shortly, the second heart field is the rotational process, and the septation is primarily neural crest, but they are connected.
Thank you. So in the chat, I think we have no further question, Prof. To conclude and to give final comments, also closing our guest professor lecture for today is our beloved professor, Professor Muhammad Sadikin. Are you still monitoring, Prof. Sadikin? I would like to invite Prof. Sadikin to give closing statement. Prof. Sadikin. Mbak
I
Adin?
think Prof. Sadikin already left
Oh, okay,
Zoom.
okay. All right then. Very well. Dr. Ria or Dr. Isabella, do you have final comments or statement for the students?
Okay. Thank you, Dr. Radiana. So I think this afternoon we have many informations, not only from me and Dr. Radiana, but many information from Prof. Marco. So I hope for students and also for our colleagues in our colleagues in anatomies and also other related lecturers can enjoy the has enjoyed these sessions, and hopefully this information have a benefit for us. So I think there's still many questions, maybe still answer question, or you want to ask, but still ashamed, for example. But I think it is a very good session this afternoon. Thank you very much. And also for Prof. Marco, thank you. You spend your time during your very tight schedules. And also Dr. Adiana, thank you very much, and other participants.
So with that, thank you very much, Prof. Marco DeRuiter. Do you have final comments for our students? Perhaps inspiring words for them, since they are still in their study phase, their research phase, their thesis research phase. Motivational quotes.
Yeah, but I think they have chosen to become a scientist. It is one of the most beautiful things that you can spend your time and your money to discover new things. And that is one of my drive to go on, and please be aware that it is always a struggle. You will never find your answers in 1 year or in 2 years. You have to work step by step with a long breath to find real clues how it works. And I hope that they will never become fatigued to study, because I think when you are deepen your knowledge, in the end you will succeed.
So thank you very much, Prof. Marco DeRuiter. This has been a very pleasant and a very productive afternoon. So I really enjoyed your lecture. They are very clear, very concise, and kind of like make me feel because the first time I entered master in biomedicine, I wanted to go deeply into heart development and stem cells. Okay, but and then I switched road and then going to the other way. Okay, so Thank you very much, students, all the participants today. I hope that this will be beneficial for your further studies, research, or your field of interest. Thank you very much. Okay, see you soon. Bye bye.
Okay, thank you, Dr. Nana. Thank you, Dr. Nana, for your excellence in moderating our webinar. So, everyone, I think we are already at the end of the event. Thank you for all of this, for our speakers, Professor Marco. Thank you very much, Dr. Ria, Dr. Isabella, for your time to share your expertise with our participants. Hopefully, the lecture and discussion today will give fruitful insight to all of the participants. And don't forget to all of the participants to join our another events. Such as in Biomedical Forum or special lectures. So stay tuned. Just check the information at our official website in biomedicine.ui.ac.id or in our Instagram at PMIB FKUI. So thank you very much, everyone. I apologize for any mistakes I did and behind the as well. So thank you. See you on next MPBS event. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone.